Hello and welcome. This will be a presentation looking at the use of the polymerase chain reaction, or PCR, in science. It will be presented by Jessica Wheel and Polly Quinn.
How does PCR work? The whole goal of PCR is the amplification of target DNA via specific primers and a constant repeat of a three-step process. Involved in these three steps is denaturation, which is separating the DNA strands, annealing, which is attaching the primers to the target DNA, and extension, so extending the DNA by the use of the enzymes. I will go through these steps in a bit more detail in the next few slides. The first stage of the PCR cycle is denaturation. This is where the target double-stranded DNA is heated to roughly 90 degrees to separate the double helix. The high temperature causes the hydrogen bonds between these bases in the two-stranded DNA to break and separate. The result of the two strands of the DNA, which acts as the templates for the production of the new DNA strands. This stage usually takes around 15 to 30 seconds, and it is important that the DNA strands are fully separated before the next stage or temperature change. Let's have a little look in the next slide of what this actually looks like. So in this slide, you can see a visual representation of denaturation, which is the first step in PCR. This is where it becomes single-stranded DNA and where the hydrogen bonds are now separated, ready for the primers to be attached. So I'm going to move on to the second stage. So annealing is when the temperature is lowered to enable the DNA primers to attach to the DNA template. The second stage is in the PCR involves the attachment of the primers and template DNA. This is done by lowering the temperature to roughly 50 degrees. This allows the primers to attach to the specific location on the single-stranded DNA template by hydrogen bonding. The primers are around 20 to 30 base pairs in length and attached to the 3' end of the DNA template. The primers are designed to create the starting point of the DNA synthesis. The polymerase enzyme can only add DNA bases to the double and strand of DNA. Only once the primers have bound can only the polymerase enzyme attach and start making new complementary strands of the DNA from the loose DNA bases. This step usually takes around 20 seconds to complete. Here's what it looks like in the next slide. So this is what annealing looks like in the second step of PCR. Here we've got the primers matching up to its base pair attaching onto the single-stranded DNA, ready for the enzyme to attach and then to repeat that sequence. Now we have the last stage in PCR, which is extension. So this is when the temperature is raised again and the new strand of the DNA is made by the Taq polymerase enzyme, so where they attach to where the primer is and then continues the base pairs along. During this last step, the heat is increased to roughly around 72 degrees to allow the new DNA to be made by a special enzyme, which is the Taq polymerase. The polymerase attaches to the primer ends and then adds the DNA bases to the single strands in the 5 to 3' end. The duration of this step purely depends on how long the DNA template is and the amount of amplification that is required. Here the next slide, we'll see a visual representation of what this actually looks like. This is what the extension looks like. As you can see, it's going from the 5' to the 3' end of the DNA. And with the dark line before it becomes spotty, that's where the primer is. And you've got all these loose base pairs that are then coming in to match up, which the Taq polymerase helps with that and continues along the strand. So the end result of PCR is that these three stages, over repeat and repeat and repeat, make a huge amount of copies of the DNA segment in a short amount of time. After the PCR is completed, a method called gel electrophoresis can be used to check the quantity and the size of the actual DNA fragments produced. As you can see here in a bit of a summary form in the picture, We've got the DNA template, the normal double-stranded double helix DNA, and then it goes through heating up, and then denaturation happens where it separates the double strands into single strands. Then we've got the 2' that come on from the 5' to the 3' end. And then we've got the extension, which is where the Taq polymerase comes on and then produces these base pairs that matches up with the DNA, which then creates a lot more copies of that specific segment of the DNA.
PCR is extremely sensitive in its ability to amplify nucleic acids. It is also able to be used on all tissues or samples, and only a very small amount of intact DNA sequences are required for amplification. It can be used on not only new samples, but also archival materials, even in poor condition. This makes it invaluable in both current and retrospective conservation and analysis of DNA. To counter some specific problems, PCR has alternative uses to overcome them. PCR cannot amplify RNA, so reverse transcriptase PCR utilizes the enzyme reverse transcriptase to allow synthesis of cDNA from an RNA template. cDNA can then be used as target DNA for amplification. Some of the shortcomings of PCR technology. The first is misleading results. PCR technology is very sensitive, and because of this, contamination of trace amounts of RNA or DNA can skew results drastically. The second is primer problems. Primers designed for PCR require sequence data, which means they can only identify or show the absence of known pathogens or genes. Primers also can, during the annealing stage, anneal non-specifically to sequences other than the target gene when they are similar but not identical. Another potential problem with primers is that they can form a byproduct known as primer dimer. This primer dimer can cause competition for the target sequences to utilize the PCR reagents for amplification. And finally, the results can be so microscopic it's tricky to interpret them. Indications of viral DNA can be so small that they are not necessarily indicative of an infection, but rather something latent or clinically non-relevant.
What is PCR used for? So PCR is used in multiple applications, among which are in genetic expression and viral quantification. PCR is also used in measuring DNA damage from diverse mutations and detect pathogens and genotyping. It has been found to be useful in diagnosing and providing direct information about various DNA by amplification. Another application field of PCR is in microbiological molecular diagnosis, the monitoring of the progression in viral diseases and measuring the effectiveness in antiviral therapies. Overall, the application of PCR is strongly used in the medical field in diagnosis, research, and effectiveness of therapies.
Future applications. Currently, quantitative PCR is still the method of preference for a variety of applications in diagnostic use. However, digital PCR, or dPCR, a variation of PCR, which provides an absolute quantification of nucleic acid concentrations, is growing in use in research settings. It offers more predictable, sensitive, and accurate results than qPCR, and along with the recent development of dPCR techniques to be more practical and at reasonable cost, it's proving to have potential for clinical diagnostic use. It's also being vetted for use in forensic applications where it has seen some good results.